everyone, welcome back to my channel. Today we'll be learning all about glycogen storage diseases and I'll show you a really nice method to keep them straight in your mind. So let's go. So first we'll go through the glycogen pathway quickly. So as you can see in this pathway that glycogen is formed from glucose and glucose converts into glucose 6-phosphate and which converts into glucose 1-phosphate, and when it attaches to UDP it forms UDP glucose and those UDP glucose then join each other to form glycogen. So you can see that the structure of glycogen that I have drawn on the right side of it, and then comes the branching enzyme and it adds some branches in that glycogen structure because glycogen is actually a branched structure. It has a branch structure and it has two type of linkages, alpha 1,4 linkages and alpha 1,6 linkages. So the subunits that are attached in a series, they are connected by alpha 1,4 linkages, while those you can see the branch points, for example, the red subunits, they are attached to the purple subunits at a branch point. This is a branch point, and here the linkage is of alpha 1,6 nature. It means that there is a connection between carbon number 1 and carbon number 6. And in alpha 1,4 linkage, there is a connection between carbon number 1 and carbon number 4. So this was the glycogen synthesis pathway, and the green arrows indicate the reversible steps of the pathway. And the red arrows indicate the irreversible steps of the pathway. So we can see that only the first two steps are reversible, whereas the rest of them are irreversible. So now let's go through the degradation pathway together. So the glycogen phosphorylase enzyme acts on that branched structure of glycogen, and phosphorylase literally means to add inorganic phosphate group without using ATP. So this enzyme adds inorganic phosphate groups to the units in the branch and helps them leave the glycogen structure in the form of glucose 1-phosphate. This glucose 1-phosphate is then later converted into glucose 6-phosphate and then into glucose with the help of glucose 6-phosphatase. You can see these reversible reactions on the top left of the screen. These are the initial reactions that we studied. So the glycogen phosphorylase acts on the units in the branch till it reaches the last four residues. These last four residues are termed as dextrins and limit dextrins, and this is where the glycogen phosphorylase stops working. So now the debranching enzyme, it acts on the limit dextrins and it is actually a transferase enzyme, and it grabs those limit dextrins, the last three of them, and places them in series with the purple units. And there is a alpha 1,4 linkage because they are now in series, so they have an alpha 1,4 linkage between them. So in this step there is only a transferase action, and the subunits, the units of the branch, are taken and placed in series with the purple units, and this is how we get this structure. You can easily see the transferred red subunits attached at the end of the purple subunits via a 1,4 linkage. So now we can see only one red subunit attached to it with an alpha 1,6 linkage. So now we need an enzyme, a debranching enzyme, which can act on this alpha 1,6 linkage. So when that enzyme acts on it, then it removes that one alpha 1,6 linkage, and that is again released in the form of glucose. Now that we have briefly gone through the pathway, we can learn about the diseases, the glycogen storage diseases, and the enzyme deficiencies associated with them. So as you can see that I have zoomed into these reactions, and you can see the top two reversible reactions where glucose is converted into glucose 6-phosphate, which is then converted into glucose 1-phosphate, and the opposite can also occur, which does occur in case of when the glycogen phosphorylase acts on the branched molecule of glycogen and starts removing glucose 1-phosphate by adding inorganic phosphates to them. They go through these steps. They form first the glucose 6-phosphate and then glucose via the enzyme glucose 6 phosphatase, and if that enzyme is missing, it is termed as von Gierke's disease, and it's a really serious disease because you have gone through all of the steps, but you cannot convert glucose 6 phosphate into glucose, and whatever the molecule is, if it's in a phosphate form, even if it's a fructose or a galactose or glucose, if it's in a phosphate form, then it is trapped inside the cell. It cannot easily move around outside the cell. It is always trapped in the cell. So the deficiency of glucose 6 phosphatase is known as von Gierke's disease, and it's a really severe disease because you can go through all of the steps of the breakdown, but you cannot go through that one final step to form glucose. So this is a very severe disease. We'll go through the clinical manifestations and the treatment in another video of mine, but in this video, I'm just going through the pathway and the deficient enzymes and the diseases associated with them. So in this slide, you can see all of the diseases that are the glycogen storage diseases. I would like you to focus on that branched glycogen structure, which can be directly converted into glucose via acid maltase, which is a lysosomal enzyme. So if there is a deficiency of lysosomal enzyme acid maltase, then it will obviously accumulate, and this disease is known as Pompeis disease. Now, I've written a big P near the arrow. It means that it is Pompeis disease. I didn't write the complete disease names because of the lack of space, but you can see the big P written near the arrow. It means Pompeis disease, which is due to the lack of acid maltase. So now we know where the branching enzyme acts on the glycogen molecule and adds all the branches. If that enzyme is missing, then we'll have Anderson disease. This is why a big A is written next to the branching enzyme. So let's move on to the next step. If glycogen phosphorylase, which has a very important role, it adds inorganic phosphate to the units present in the branch, and it acts on them and removes them till it reaches the end of that branch and only the dextrins remain. Dextrins are 3 or 4 residues which are remaining on the branch after the glycogen phosphorylase has acted on them. So if this glycogen phosphorylase is missing, we'll have McArdle's disease, specifically the glycogen phosphorylase present in muscles. So that disease would be called as McArdle's disease. So now we only have the steps in which the debranching enzyme acts on the remaining residues and removes them and transfers them and removes the 1,6 linkage, and we get a glucose molecule. So if that debranching enzyme is missing, we'll have CORI's disease. There is also another disease which is, I believe, called Hirsch's disease, which is due to the deficiency of hepatic glycogen phosphorylase, but it is not very high yield. So I would not suggest you to remember that. So these are all of the diseases that are associated with this pathway, and these are collectively termed as glycogen storage diseases. So I know it's a lot to process and it is not very easy to remember all of this. So I'll show you how I remember and keep all of them straight in my mind because it is very important and these are very high yield. So the sentence I use to remember all of these diseases and the associated deficiency of enzymes is von Gierke's personal assistant may mumble ABCD. So, as you can see, von Gierke's, I have highlighted the G from von Gierke. So, it has deficiency of glucose 6 phosphatase. So, now, personal assistant. Personal assistant, the P, which is highlighted in green, stands for Pompeis disease, and assistant stands for acid maltase. The A in assistant stands for acid maltase, since Pompeis disease is due to the deficiency of acid maltase, which is, if you remember, a lysosomal enzyme. May mumble. The M in May stands for McArdle's disease, and the M in mumble stands for myophosphorylase. It means the glycogen phosphorylase present in muscles. So, A, B, C, D. What does these mean? A stands for Anderson's disease, and B stands for branching enzyme, C stands for Corey's disease, and D stands for deep branching enzyme. And because Anderson disease is due to deficiency of branching enzyme, and Corey's disease is due to the deficiency of deep branching enzyme. So you just have to remember one sentence. Von Gierke's personal assistant may mumble ABCD. So you only have to remember this sentence and you will never forget the glycogen storage diseases and their associated enzyme deficiency. Hope you found this video helpful. Don't forget to like, share, and subscribe. Hope you have a great day. Take care of yourselves. Bye bye.